Hi, everyone. So I am especially excited to introduce Dr. Giles Yeo, who for the NHGRI DIR seminar series. I personally have been following his work and his science communication for many, many years. I may be the unofficial head of his NIH fan club, I'm not sure, but I would take that position. This is all for really, really good reason. So Giles Yeo is a professor of molecular neuroendocrinology and program leader of the MRC Metabolic Diseases Unit in Cambridge, a fellow of Wolfson College, and honorary president of the British Dietetic Association. He was appointed as an MBE in the Queen's 2020 birthday honors for services to research, communication, and engagement. I had to look this up. It means that he's a member of the most excellent order of the British Empire, which sounds very impressive. He won the Society for Endocrinology Medal in 2022, which I know is impressive. And as I mentioned, he's a very prolific science communicator, presenter of science documentaries for BBC, podcast host, author of 2 very good books. I won't go on, but I will just remind the audience that this is hybrid. So if you are online, please submit your questions in the Q&A on Zoom. And if you're in the room, please use the mic so that those online can hear you. So without further ado, I will bring you on up to start the talk.
Thank you very much, Susan, for inviting me. I am glad I wasn't sent home, and that's all I'm saying about it. So, folks, so my name is Giles. Thanks. I'm based at the University of Cambridge, and I'll be here talking about mapping. Well, actually, I'll be here to talk about the genetics of body weight. But I'm beginning this talk slightly differently. There'll be 2 parts to this talk. The first will be my efforts, or our efforts, in mapping feeding circuits within the human brain, and we can go with that. Then there'll be a pretty hard left turn, and you'll see that inflection point pretty clearly when I then go from there into genetics, but then using as well this resource that we've created in order to do things. And so look, I'm a geneticist by trade. And I think being a geneticist is a perfectly upstanding thing to do. You know, my mother-in-law still speaks to me, so yay. But when people ask what I study, and I say I study body weight, on one end of the spectrum just happens to sit obesity, immediately I become the bad person. And I become the bad person because I'm perceived as giving fat people, overweight people, people living with obesity, terms I do not use in any pejorative fashion whatsoever, and excuse, which philosophically has always been a very interesting take for me. Because if I was studying the genetics of pretty much everything else, okay, cancer, dementia, arthritis, whatever, you know, would I suddenly be giving those people suffering from those conditions an excuse? No, I'd be trying to understand biology, mechanisms. I mean, God forbid, I might be trying to help somebody. But when we talk about body weight, suddenly it's infused with choice. And so this mishmash of a lecture, which I've kind of a talk, which I've put together, hopefully there's some element of a rebuttal within there. So mapping the feeding circuits in the human, in the human brain. So we work on this a general schema in which we try and understand the brain control of food intake. And your brain needs to know, broadly speaking, 2 pieces of information in order to influence your food intake, internal pieces of information. It needs to know how much fat you have, your long-term energy stores, adipose tissue mass, which is how long you would last without any food. And it needs to know what your short-term energy stores. And this is going to come from your gastrointestinal tract. All of these are hormonal. These long and short-term signals are hormonal. They circulate in the blood, clearly, and then signal to a part of the brain called the hypothalamus. For those of you who don't know where the hypothalamus is, bridge of the nose, base of the brain, sitting right above the pituitary. In human beings, is about the size of your thumbnail, shape of an almond. And within the context of body weight, because hypothalamus does many things, but within the context of body weight, it acts as a fuel sensor. So it acts as one of the key areas in the body which senses these long and short-term signals. Your brain then responds and translates these signals and then influences your next interaction with a menu, with a refrigerator, or with a restaurant. And as I said, there are genetic modifiers that run throughout this process. And we now know that the genetics of body weight is, by its very definition, the genetics of how our brain influences our feeding behavior. Okay, so that is where we're actually coming from. Now, I'll be focusing on one part of the brain, the hypothalamus, which I said, but the hindbrain also performs some elements of this of the same effect. So this is a schematic of the hypothalamus over here. It's obviously a squashed schematic of it. And I highlight this because it's probably the most heterogeneous part of the brain, so that's interesting in of itself. But I've also drawn, specific neuronal populations here, like POMC neurons and AGRP neurons. And I highlight this only to say that we have a tendency when we draw, and I drew this figure, when you draw these figures like that, where you have a tendency to sort of think that these are homogeneous groups of neurons, which they're not. Because I think within these POMC neurons, AGRP neurons, whatever neurons, there is a lot of heterogeneity, therapeutically relevant heterogeneity, I want to argue, that sits within there, which is of interest to us to actually study. And so we use single cell genomics as well as spatial technology in order to try and unpick the heterogeneity of the hypothalamus. We began in the mouse, and this is published, so I'm just showing this one thing where what we did was using our own data as well as pooling together 17 or 18 of the largest at the time single cell work in mice. We sort of integrated all the data so that we could actually use it as a comparative database in order to do so. And so we ended up on the cover, which is always a good thing. A couple of things though about this. This is version 1, and so this is version 2 is coming up soon. Hypermap 3D we call it. But it says mouse hypothalamic atlas on the journal cover, and I'm not complaining, but I am complaining. A couple of things. That's not a mouse brain. That's the first problem, and that's not the hypothalamus. So entirely inaccurate. But I didn't pick the figure, the picture, and you can go to the QR code, and for those of you who care about this, and actually play with the data. But a big reason for us doing this initial work in terms of mapping the mouse hypothalamus is to sort of use it as a testing ground for the algorithms and technologies we were going to need to do for our prime project, which is to do the same thing within the human hypothalamus. Now, the human hypothalamus, for obvious reasons, is quite difficult to get to. You can't get to a human brain legally, from a living being. But now with obviously the single cell and spatial technologies, as well as access to human brain donors from brain banks, we're now able to actually put this together. So we've used the MRC Brain Bank network for this, but recently we've now got access to the NIH Brain Bank as well, and so we'll begin to get brain samples as well from the Brain Bank. This Brain Bank network, I think is a rather dispersed center for that. And so here is the human our efforts, okay, which has just gone in press and should be out hopefully in a few weeks. And you can see here a UMAP for those of you who work with single cell data. This is what this is, and we have divided it up from non-neuronal cells and neuronal cells that are here. And then there's a circular dendrogram, a circle of life, shall we say, in which we can actually then see the various neuronal populations. We've only given the first 4 levels here, so there's actually more visual, but it actually goes up to 6 levels, and there are 450 or so different types of population. 40% neurons, 60% non-neurons from there. So if you then, the first thing we did was to actually compare the human database to the mouse database, okay? Because yes, we use mice as a model, but how similar, because it's one of these things, right, where the vast majority of feeding circuitry at the moment, our understanding has come from mouse work, and we are very, very good at now removing specific genes from specific neurons at specific times. But how many of these populations actually exist in the human context? And so this pretty diagram over here, broadly speaking, it's most of the populations that matter do exist in both humans and mice, broadly speaking, which is why we use mice as models, and that's absolutely fine. But what I want to, and I'll give you one specific example later, there are relatively nuanced but therapeutically relevant differences that are well worth actually trying to understand when we're looking at these, and I'll highlight one of those in a bit. And so we also use spatial transcriptomics in order to try and understand this. And so this is a few brain slices, and you can see here, for example, you can see the spatial markers. Here's the arcuate nucleus. Here's the ventral medial hypothalamus. Here's the paraventricular nucleus. Not a lot there because it's a different slice. And then here you can see the reasons why spatial transcriptomics is important. These are glutamatergic neurons. These are GABAergic neurons. And you can see that there is a enrichment of glutamatergic neurons within the ventral medial hypothalamus, whereas GABAergic along the outside. So there are always spatially distinct things that we can actually tell from here. Now, that was not, at the time when we did that work, it wasn't single cell level. Okay, now you can get single cell single cell spatial transcriptomics. And so we needed to sort of put the single cell stuff together with the spatial stuff together. And so we used a Bayesian approach called cell to location, which is written by the Biorector Lab from the Sanger Institute, in order to deconvolute the single cell data into the spatial data. And so this is what it looks like. We've taken just the ventral medial hypothalamus, and at level 3, we find five different clusters of cells, of neurons, and this is what it looks like when you actually map it from a spatial perspective. They all map within the ventral medial hypothalamus. That's because it's not surprising, that was what we're choosing it. But you can see that each of the five, and they're color-coded, each of the 5 different clusters here map in different. So the blue one, for example, maps here, whereas the dark blue here, and you can see spatial distinction. Now, this is just a pretty picture for now, obviously, but this is whole transcriptome, which means that you can ask any questions you actually want. And from here, you can then begin to actually ask some useful questions. So for example, now this is the hottest thing since hot things. These are the new anti-obesity drugs that are actually around. Tirzepatide, or semaglutide, which is either Ozempic or Wegovy, and tirzepatide, which is either Zepbound or Mounjaro, and they target the incretin receptors. So if we actually then map the incretin receptors, where do they actually sit? Certainly within the human hypothalamus, right? Because all of the previous work about mapping this have all come from mouse, have all come from the mice. And so if we then do that, let's focus on the GLP-1 receptor. So GLP-1 receptor is the receptor for semaglutide, so Ozempic and Wegovy. So first piece of information is that the GLP-1 receptor in the brain, at any rate, because obviously there are also peripheral receptors, in the brain is exclusively neuronal. Okay, so that's the first piece of information. And then we can then have a look to see, well, where within the hypothalamus and what spatial relevance is there. So here, for example, is something within the paraventricular hypothalamus, and because it co-expresses SIM1. It is interestingly the only part of the hypothalamus that also co-expresses the GIP receptor. So GLP-1 receptor is for semaglutide. Tirzepatide targets both the GIP and the GLP-1 receptor. So this is the only part of the hypothalamus that is co-expressed. Everywhere else, they're single operators. They are completely separate sets of neurons. And if you see where they map from a spatial perspective, they map within the paraventricular hypothalamus. Now, how about then within the arcuate nucleus, so another part of the hypothalamus? So here we see 2 distinct types of populations of the GIP receptors. Ones that co-express the leptin receptor and POMC. I'll come and talk about these in a second. And ones that express the calcitonin receptor. And these then map in completely different areas. They all express the GLP-1 receptor, but they map in completely different areas. So for example, the ones that co-express leptin receptor and POMC are expressed down here, whereas the ones that express the calcitonin receptor are completely different parts of the hypothalamus, wrapping the ones with the leptin melanocortin pathway. Now this is relevant because at the moment, Novo Nordisk, who make semaglutide, are testing combinatorial therapies, including semaglutide together with their calcitonin receptor agonist, cagrilintide, called CagriSema. So it is important to know, particularly where the cells expressing GLP-1 receptor, calcitonin receptor, and where they're actually co-expressed as well. Now here is probably one of the first species-specific difference that we actually find. So in human beings, if you actually, in mice, let's do mice first. In mice, if you have a neuron that has GLP-1 receptor and leptin receptor, and then there are separate populations of cells which express POMC and the GLP-1 receptor. In humans, and this is in mice, there's separate populations in mice. In humans, if you have the GLP-1 receptor and POMC, you always express the leptin receptor as well. So there are a group of, a single group of neurons, relatively nuanced, but given that they're drugs targeting all these cells within the brain, I think it's important to actually understand what they're expressed with. Now, how about the GIP receptor? Now, the GIP receptor, unlike the GLP-1 receptor, within the brain is not exclusively neuronal. In fact, it's mostly non-neuronal, 60% non-neuronal, 40% neuronal, okay? And largely, they're expressed within oligodendrocytes. So this big lump of cells over here, So of the 480,000 cells, or 450,000 cells, 200,000 are oligodendrocytes. 10% of them express the GIP receptor. What the hell are they doing there? No clue. Okay, we have no idea what they're doing there at all. And if you look at the neurons, they're not particularly interesting. They're there. They're expressed within neurons. So I'm not going to show you any pretty pictures. But if you look at the non-neuronal cells, then the oligodendrocytes have spatial distinction. That's fine. We're still trying to figure out what the hell they're doing there. But here is a really interesting population of cells, the ependymal cells, okay, which also express the GIP receptor. And you can see that where they actually sit is they line the wall of the third ventricle of the hypothalamus. It's very, very, very specific. Now, Mounjaro, tirzepatide, is more effective, subtly more effective than semaglutide. People have been wondering why. Is it because it targets both incretin receptors? So in other words, is it dual agonism? Or does the binding of the GLP-1 receptor here enhance access of the drug to the whole hypothalamus? I don't know the answer, but now at least because we know where they're expressed, we are in a position to actually be able to at least ask the questions that were there. Okay, and I can show you any number of other things, but I won't bore you to death. So genetics turned now, okay, where we then decided to try and map the genetics data onto these neuronal clusters or the cellular clusters. And so if we actually take GWAS genes first, and there are over 1,000 GWAS loci now. So if you take the locus, the nearest gene to the locus with the BMI GWAS hits, and map and ask which of the 452 hypothalamic clusters, if any, are they actually enriched in. And we actually find that 291 of these cell types are enriched for these BMI GWAS genes. And if you see where they map, and on the left-hand side, what you can see here is you can see where they are within hypothalamus, neurons or non-neurons, and you can see that the enrichment is exclusively within neurons. So while I say that the genetics of body weight is the genetics of how our brain influences feeding behavior, it's not just a geographical area, it is specifically a neuronal signal that we see. Okay, so that was interesting in of itself. Then what we asked was, okay, well, of these 1,000 genes, which were driving the enrichment? And when we did that, then we found that the enrichment were not driven by all 1,000 genes, but were driven specifically by 426 genes. So now we have this. We then did a gene burden analysis, an XWAS, okay, to actually find whether or not rare changes within the coding regions of these genes have influence on population level changes. So just briefly what an XWAS is. GWAS, clearly you take an individual signal, you ask whether or not it co-segregates with a specific trait. If it's 50-50, then it's random. If not, and you take each individually. And it has to be common enough to appear on an Illumina chip or what have you, all right? The difference, and so it has to be common, and it's largely non-coding because 98% of the genome is non-coding. But an XWAS is what happens is you do whole exome sequencing at scale. So we're looking now at the whole of UK Biobank, for example, 500,000 people has been done. And now you can look at the coding regions and look at all the changes, rare or common. Now, it's called an XWAS, or a gene burden analysis, because instead of each individual signal being the statistical moiety you're testing against, what you do is you aggregate all of the changes within a given gene. So the gene has become the statistical moiety. You then aggregate all of the changes, rare or not, into it. It's a gene burden, and ask changes in a specific gene, loss of function changes, is it associated with body weight change at the population level? Okay, that's what an XWAS is. And we found 6 of those genes did reach population level significance. Now, a few of them are positive controls. Okay, the MC4 receptor, PCSK1, the calcitonin receptor. Where is the signal? The calcitonin receptor and POMC. So these we have known are actually at the population level. But we also identified a couple of new genes that I want to talk to you about. So I'm going to focus now and actually change the tenor of the talk entirely to go to a purely genetics talk, where I'm going to focus on the melanocortin pathway. I'll talk about that in a second. And then I'm going to end with this new gene we identified called bassoon. Okay, and that has population level changes in terms of body weight. So now the leptin melanocortin pathway. So for those of you who don't know what that is, this is the fat sensing part of the brain. And so leptin is a hormone, it's an adipokine that circulates in proportion to the amount of fat you have. So the more fat you have, the more leptin you have. and it signals to the hypothalamus, as I say. Within the hypothalamus, leptin signals to a population of neurons called the POMC neurons, or pro-opiomelanocortin. POMC is sliced and diced by prohormone convertases into biologically active peptides, melanocortin peptides, hence the leptin-melanocortin pathway, which signals to the MC4 receptor, the melanocortin 4 receptor, and 3 receptor. I'll touch on that briefly as well, in order to influence body weight. Now, for this part of the talk, there are a couple of narratives. So first is that all mammals and any higher vertebrates that has a demonstrable fat mass will have a leptin-melanocortin pathway, because this is how we sense fat. Okay, so that's unsurprising. It's a conserved pathway. What was surprising, and still is, I think, to those of us within the field, is that genetic disruption of the pathway as a strategy to influence feeding behavior is also conserved across. And so I'm going to highlight just a couple of some traditional but some surprising animals that have naturally occurring mutations within the pathway, which actually influences their feeding behavior. So POMC, okay, first of all. So where is the first genetic evidence that POMC results in severe obesity? So this is work actually done by my colleagues at the Charité Hospital in Berlin, Annette Grutus, Heiko Kruder. And POMC is a complex molecule because it does any number of different things. It signals to 5 different melanocortin receptors. And you can see that if you have a human POMC mutation, this is a homozygous mutation this child has, is that he has bright red hair because of a lack of melanocortin action at the melanocortin 1 receptor, which is important for skin coloration and hair color. So natural redheads will have homozygous mutations within the MC1 receptor, making their head bright red. Okay, it's a recessive disorder, but actually, trait. But actually, 40 to 45% of white Northern European Caucasians will be heterozygous for polymorphisms within the MC1 receptor. And you know the phenotype because you're unable to stand in the sun without factor 500. Okay, so you can't, there's no protection at all from the sun, heterozygous for the MC1 receptor. It's got isolated ACTH deficiency because it doesn't have ACTH, that's a product of POMC, and it's got hyperphagia and obesity because of a lack of melanocortin action at the MC4 receptor. So we knocked out the mice in 2000 in order to understand the biology, but we've also been more recently working on Labrador retrievers. Now, I don't know if there are any Labrador owners in the crowd over here, but it's the most popular pet dog in the UK and I think North America as well. And in the UK, we use them to sell toilet paper. It's the Andrex puppy. Now, and this is work that was led by my veterinary surgeon colleague, Eleanor Raffin. Because these dogs are tremendously food motivated, very food motivated. You got to keep all your compost bins shut, everything, because they'll be in there eating everything there. And what is interesting is that a percentage of Labradors have this deletion in POMC. Okay, so they have a sort of broken fat sensing pathway. Now, not all the Labradors have it, and so when you do a genotype-phenotype correlation, and what you see here is weight in kilograms, you can see a gene dosage effect. So the heterozygous Labradors are on average 2 kilos heavier than the wild types, and the homozygous on average 4 kilos heavier. And that doesn't sound like an awful lot, but Labradors only get to around 30 to 35 kilos, so 4 kilos is a lot of dog. Now then, when we did food motivation, and in this particular situation, the food motivation was done by questionnaire to the owners, not the dogs. But actually, they're modified feeding behavior questionnaires for parents, okay, to their children, because some people consider pets as their children, but modified questionnaires, and it works. And you see exactly the same pattern over here. And the interesting thing about studying food behavior in dogs, I'm not a doggy person, but obviously Eleanor's a vet, is that you can't use a traditional buffet, ad libitum test meal. And the reason you can't do that is because dogs, in particular Labradors, don't actually know when to stop. So if you've given them, they will really literally harm themselves. So at the moment, what Eleanor does, for example, one of the studies, in addition to questionnaires, is there is a sausage in a cage study. A sausage in a cage, as you might imagine, you have a sausage, you have a cage, and you can't get into the cage without opposable thumbs, okay? And so the dogs don't. And so the experiment is, how long will the dogs focus on the sausage in the cage? A bloody long time is the answer, okay, when it doesn't, and so that's some of the studies that we're actually doing. Okay, so this is what I was saying, a couple of kilos per deletion allele, and 0.3 of a standard deviation of body weight. For those of you who follow the BMI GWAS studies, 3 times the effect of FTO. But this is not the end of the story. A couple of reasons why. First, only 20 to 25% of pet Labradors have this deletion. But 95% of pet Labradors are really food motivated. So Eleanor right now is throwing the genetic book at this, omics and WAS and whatever at it, okay, in order to try and find it. She's got a paper now actually in Press at Science in which we've actually, she's identified a few more genes as well. So point number one. Point number two, there's a denouement to this particular tale. So pet Labradors have a wonderful temperament, which is why we have them as pets. But Labradors are also very, very, very trainable. In fact, they're so trainable, they're primarily used as assistant dogs, guide dogs to the blind in particular. And guide dogs are like the Navy SEALs of the dog world. I mean, they've been trained within an inch of their life because they're about to be given a human being to look after for the rest of their life. And the training pyramid is very steep. Loads of failure, nothing happens to them, they become pets. But you get the few, the proud, the guide dogs. And they're trained, interestingly, using Pavlovian approaches with food, okay? 80% of successfully trained guide dogs have this deletion in POMC. So here's the concept, okay? Imagine if I'm the guide dog and I'm bringing home visually impaired Mr. Smith, okay? And what happens if suddenly a chicken runs across the road? Now, in doggy algorithm, what are the chances of having chicken for dinner? Is it 50-50? Is it 80-20? It doesn't matter because the successfully trained guide dog understands that it has a 100% chance of getting dinner if he brings Mr. Smith home safely because of the selection pressure that's actually applied upon there. So we ended up also on the cover of this particular journal. The human being is Eleanor, who still leads the study. And Jasper is the chocolate lab that's there, one of our participants. So for those Labrador owners who go back home and the dogs have big googly eyes and said, "Oh, Jasper loves me." He doesn't love you, he's hungry. It's true. I'm just telling you the truth, ladies and gentlemen, just telling you the truth. So this is where I actually ended up in terms of from a chronological order where I ended up on the scene. This was my first paper out of my postdoc. And we identified the mutations within the MC4 receptor, which POMC binds to. So we're now working down the pathway. POMC binds to the MC4 receptor, results in severe obesity. But here is where I think it gets interesting. Now, it's interesting enough that the next few slides I'll focus on the MC4 receptor. And there are a number of interesting things about the MC4 receptor. So first, it doesn't only make you eat more, it actually changes your feeding behavior. So this is not work that's done by me, it's done by my colleagues. So this study over here uses a chicken curry eat and mess study. I'll explain it in a second why. All right? So first of all, the study participants. You're gonna have people with mutations in the MC4 receptor, all heterozygous mutations, so therefore have severe obesity. You have body weight matched individuals, but without the mutation in the MC4 receptor, and then you have controls. Now the study is you walk in, and let's start with the curry study. There are 3 trays of curry. Now, why curry? Because you can adjust the amount of fat that goes into the food by whisking in vegetable oil, all right? Now, it won't change the flavor, but it might change the texture, and some people like greasier food, other people don't. It's a personal taste. So you come in, all 3 groups, you taste all 3 curries, you rate the one you like the most, and then you eat the one you want. So as you might imagine, there's a distribution, but all of the people with MC4R mutations go for the high-fat option, all of them. So were we surprised? Well, we're thinking, well, we know that mutations in the pathway make you eat more. Maybe one of the mechanisms is by making you eat more energetically dense food. Then we get to the eat and mess study, which was interesting. So what is eat and mess? Eat and mess is what happens when you take a pavlova, drop it onto the floor and scrape it back into a bowl, all right? So it's an eat and mess. But what happens there is you can adjust the amount of sugar in eat and mess, in the whipped cream, and in the meringue. So now you reset the study up, but now with a Goldilocks selection of the amount of sugar. And here's the interesting thing. The mutations, the carriers of mutations in the MC4 receptor all went for the lowest sugar option. So now one mutation, one gene, and suddenly we like high-fat food and the low sugar option. Here for that. So it doesn't only make you eat more, it changes your feeding behavior. Interesting fact, number 1. Interesting fact, number 2, it's more common than we thought. Okay, so we were studying it as a rare condition within severe onset obesity, but then we began to realize In true severe obesity, so 99.9 centile, we were probably finding it anywhere from 6 to 10% of the population, so really quite relatively high, but in severe obesity. But what happens in the general population? So this, we worked with our colleagues in Bristol, who work with the Avon longitudinal study of parents and children, all right? And it's Avon because of the river Avon, which actually comes out by Bristol. And this is a birth cohort rather than a disease cohort. So in other words, we didn't go out and find severely obese people. Women pregnant between 1990 and 1992 were invited to come to the study in Bristol, and then their offspring have been followed up since then. So now they're in their 30s, obviously. Colloquially, it's called the Children of the 90s study, but it's called ALSPEC. It's 12,000 participants. So we screen, and it's an all-comers, okay, so anyone there. So we screened the MC4 receptor in this cohort and found that 0.3% have loss-of-function mutations in the MC4 receptor. So if you extrapolate that to the UK population, 66 million people, that's 200,000 individuals, all right? If you extrapolate that to the US population, that's more than 1 million people with mutations within the MC4 receptor. And there is no good reason to imagine why there are differences, certainly in these rare loss-of-function mutations rather than SNPs, in prevalence in different populations, because we found MC4 mutations in pretty much every population we have looked for to date, and as you'll soon see, in most creatures as well, all right? So 0.3%, okay, which is a lot of people. So what does it look like? What happens in the wild, okay? So at 18 years old, because you remember this is a longitudinal study, at 18 years old, heterozygous mutations in the MC4 receptor mean that you are, on average, nearly 18 kilograms heavier. So 40 pounds, okay? That's a lot of weight, heterozygous mutation. This 18 kilos average is, roughly speaking, 5 BMI points. So it's a lot of BMI points, 20 to 25, 25 to 30. And of that 18 kilos average, 15 kilos is fat. 200,000 people, more than 1 million people within this country potentially having mutations. Interesting point number 2. Interesting point number 3, there is now a drug that targets the system. So this is the same group that identified the first kids with the bright red hair for POMC deficiency. And they worked together with the pharmaceutical company, Rhythm Pharmaceuticals, with an MC4R agonist, a drug called setmelanotide. Okay, so this is the original paper, the proof of concept paper. It's all color-coded, so just look at the red growth curve that we actually have here. And the study here is setmelanotide, so daily injections, which binds to the MC4 receptor in two kids with no POMC, homozygous POMC mutations. And just to give you an idea of scale, so patient 1 in red, at 18 years old, patient 1 is 160 kilograms, all right? So that's what, 350 pounds, there and thereabouts. So these are big people, okay? So, and then patient 1 is treated. So patient 1 is treated for 42 weeks. So the x-axis is weeks, the y-axis is weight in kilograms. And what you can see here is after 42 weeks of treatment, patient 1 has lost 50 kilograms, 50. Now the longitudinal data is now out, just came out a couple of years ago, same color coding. Look at the red patient 1 again, and the x-axis still remains weeks. So we see now that it's gone out to 7 years, 350 weeks or so. Couple of things. You can see that the maximum weight loss is achieved around the 2-year mark, 110 weeks or so. 70 kilograms has disappeared. A whole of me has disappeared from this human being because of daily injections of setmelanotide. And so this drug, so there's a drug that targets the system and has really energized the field. So this drug is now approved by the FDA for now. Has now been approved for rare genetic causes of obesity, so POMC deficiency, PCSK1, which is the enzyme that chops up POMC deficiency, leptin receptor deficiency, Bardet-Biedl syndrome, and a couple of other rare disorders, and it's currently being trialed and tested for MC4R deficiency. I don't think it's going to be a Ozempic blockbustery thing. It doesn't appear to work particularly effectively in generalized obesity, but certainly when it comes to specific genetic causes of obesity, this is actually a well worked out system now, okay? Interesting point number 3. The last interesting point is if you think about 0.3%, it's not super rare, it's not super common, but you know that evolution is pretty damn good at stripping out a change that has negative consequences, no matter how small, to your reproductive capabilities. So it's kept it around for some reason. What happens when we look at other species? Okay, so for example, let's look at the livestock industry. Okay? Now these pigs, not that pig's from Google, but the pigs within this particular paper here are bred for bacon. Okay, that's just what they are, they're bacon pigs. So what are livestock farmers, what are they doing? Okay, well, they do selective breeding, right? You pick specific traits that you want from growth trajectory, fat to lean mass ratio, whatever it is, and then you do selective breeding. We've done this for millennia. This is how we domesticated animals. All of these pigs have mutations in the MC4 receptor. And these are not genetically modified pigs, naturally occurring mutations, which because of selection pressure has then been enriched. All of them have mutations in the MC4 receptor. And this is not some weird mammalian thing, neither. Okay? So let me introduce you, ladies and gentlemen, to the blind Mexican cavefish, which really begs two questions. Why are they blind? And why are they Mexican? I'm gonna tell you the answer. So the big asteroid that came down and killed all the dinosaurs 65 million years ago hit what is now the Gulf of Mexico, or maybe the Gulf of America. Anyway, so into the Gulf of Mexico, and when it hit the bottom of the ocean, cracks formed. They had underwater caves. Fish got stuck in them, hence they are Mexican cavefish. They're blind because there's not a lot of light. There's no light in an underwater cave. So over millions of years, their eyes evolved away. Why am I telling you any of this? Because there's also not a lot of food in an underwater cave. So any Mexican cavefish who was remotely blasé about the plankton that was floating by and didn't snap it up became an ex-fish. Every single blind Mexican cavefish, and there are a lot of them, okay, have mutations within the MC4 receptor. Ladies and gentlemen, where is the choice? Okay, people think feeding behavior is a choice. I'm talking about a fish, pigs, Labradors, 1 million people potentially in this country. Okay, where is the choice? If you supply enough of a selection pressure on an existential trait, and really there are only two true existential traits, reproduction and feeding. You fail at one, you fail at life. We understand this. So if you put a powerful enough selection pressure on an existential trait, it begins to hardwire behavior, including feeding behavior. Where, ladies and gentlemen, is the choice? Now let me briefly segue into the MC3 receptor. Now there are 5 melanocortin receptors, 3 of which are peripheral, 2 of which are central in the brain. The 2 central receptors are the MC4 receptor, which I've just talked to you about, and the MC3 receptor, melanocortin 3 receptor. They're all through genome duplication, so they're all homologs of each other. Now the MC3 receptor was not a straightforward story. And we were studying it at the same time we started studying the MC4 receptor, which was the late 90s. And equivocal, okay? You would look at all of the studies done prior to this paper I'm gonna tell you about, and actually, it was a 50-50 thing about whether or not the MC3 receptor had to do with body weight or not. So now, however, because we are in an era where I want to argue that genetics has been democratized. What do I mean by this? Where you actually have the large cohorts, UK Biobank, any number of other cohorts have now been sequenced, whole genome sequenced even, where we can now just ask the question about these. Now we used 3 different cohorts, which are probably worth telling you about. We use LSPAC, which I already told you about. This is the longitudinal data, but relatively small, 12,000. Okay, UK Biobank, 500,000 healthy UK controls. Okay, that gives you power because of it, but it's cross-sectional. And Genes and Health. Now, Genes and Health is an interesting conglomeration of 4 different cohorts. It's a study, 50,000 individuals, a study in the UK which focuses on UK South Asian, people of South Asian descent from the UK. In particular, people of Bangladeshi and Pakistani descent. So in London, for example, is East London Genes and Health, which is roughly 50-50 people from Pakistani descent or Bangladeshi descent. Born in Bradford, which is in north of England, which is pretty much exclusively from Pakistan, and a population in Manchester and a population in Birmingham, 50,000 participants. Why these populations? These separate studies were originally set out as, because they were immigrant-based populations, as a sociological study to see insecurity and education, what have you. And it then became a more broader genetic study, but because there are increase of consanguinity, so cousin-cousin marriages within these populations, we were then able to identify rare homozygous loss-of-function mutations. So LSPAC for longitudinal data, UK Biobank for power, Genes and Health for rare homozygous loss-of-function mutations, which were able to harness all 3 to understand the biology of the MC3 receptor. So first, UK Biobank. So for UK Biobank, we just said, okay, where are all the mutations within the MC3 receptor? We functionally characterize them and look for the loss of function mutations. And we're now in a position to understand what happens when you don't have the MC3 receptor. So I've color-coded the phenotype in the heat map. Green is increased, purple is decreased. And when you have a heterozygous mutation in the MC3 receptor, you have a delay in puberty. Age of menarche in girls, age of voice breaking in boys. Now, how much of a delay? Now, this depends on the level of dysfunction. If you look at this particular mutation over here, F45S, it is a dead MC3 receptor mutant. Okay, we've tested it, completely dead. 1 in 700 carriers in UK biobanks, so we found over 1,000 of them. If you have a heterozygous mutation, half a year delay in puberty. Now remember, these are not kids that are in a puberty clinic. These are just regular, normal people, normal distribution. Half a year is a pretty long time for delay in puberty. Now, if you then focus on this particular mutation up here, V44I, now that is found in 1 in 10 UK biobank carriers, so if we want 50,000 carriers, delay in puberty, and it's close to wild type in terms of the function, and in terms of the delay in puberty, 3 weeks. But because we have 50,000 individuals, we're able to actually pick this deuce down. So delay in puberty. This delay in puberty is associated with a reduced somatic growth, okay? So this includes adult height, sitting height, leg length, total lean mass, as well as reduced IGF-1. But crucially, if you look at all of the anthropometric traits to do with obesity, so BMI, waist to hip ratio, risk of type 2 diabetes, entirely white. Okay, so within the context of population genetics, the MC3 receptor, while associated with timing of puberty and somatic growth, is not an obesity gene. We found one, at the moment we have more now, but we found one rare homozygous loss of function mutation, dead receptor, so it's a null, okay, and with one person from Genes and Health, and it's a G240W mutation found in an individual of Bangladeshi origin, completely dead receptor. And he displayed exaggerated phenotypes compared to heterozygous carriers. For example, he did not go into puberty till he hit his 20s, which is pretty late. But crucially, he's not infertile because he has 3 kids. So the MC3 receptor influences the timing of puberty, but is not required for puberty. So how might this be happening? Well, so what we did here was we then went back to our single cell data, and you can see where these are. So I've mapped, just for context, I've mapped where POMC, the ligand for the melanocortin receptors, sits. We found 3 different populations of POMC, ones that express the leptin receptor, ones that express calcitonin receptor, and ones that express SOX3, and you can see that they sit in different parts of the hypothalamus. Now look at what happens when you actually look at the MC3 receptor, and first of all, you can see a completely different pattern of expression. And there are 3 different populations of MC3 receptors. We see MC3 receptors expressed on kisspeptin neurons. Now, kisspeptin and its receptor, GPR54, is required for puberty. If you have mutations in either kisspeptin or its receptor, you're infertile. So it does appear that MC3 receptor on kisspeptin neurons influences the timing of puberty. It's co-expressed with GHRH, growth hormone releasing hormone, neurons, and we think that this is where it's mediating its growth phenotypes. And so this is what we think is happening. So where are we with the central leptin melanocortin pathway? And so we have a model, this is a model, of we think a bifurcating model of nutrient sensing by the central leptin melanocortin pathway. So what are the secular trends in human biology today? Well, we are certainly getting fatter, we are getting taller, and we're going into puberty earlier. And it doesn't matter which populations we're studying, this is pretty much true at all the different populations. And we think that this is being mediated, at least in part, by the central leptin melanocortin pathway. So you have these signals, long-term energy signals coming in, leptin, as I talked to you about, insulin, which I haven't talked to you about today. And then via the melanocortin neurons, via the MC4 receptor, regulates appetite and energy storage. I've talked to you at length about that. But via the MC3 receptor, regulates growth, accrual of lean mass, and the timing of sexual maturation. Now, I'm not talking too long. Okay, just a few more minutes. And so I'm gonna end on bassoon. So bassoon is one of these new genes that we identified using this gene burden analysis. 2 different gene burden analysis, one from the hypermap study I just showed you, but one through a study which I did with my epidemiological colleagues. So bassoon is a or BSN, is a presynaptic protein with a role in exocytosis-mediated neurotransmitter release, and it's called bassoon because it's bound to piccolo. because scientists think we have a sense of humor. But I think the deep disappointment for me is if you're going to have a metaphor, well, lean into it. Where's the rest of the orchestra? I don't see no clarinets. Where are the violins? Anyway, it's just bassoon and piccolo. So anyway, so what we then did was, this is based on actually inspired by a study, and this is the first example from Regeneron, Akbari et al. In Science in 2021, so relatively recently, the first example of this gene burden analysis, this XWAS at scale. So they sequenced 640,000 exomes, including the whole of UK Biobank, as well as a number of other studies based here in the United States, okay? And identified a number of genes that I described to you using that gene burden analysis that are novel and are linked to obesity, okay, for that. And so our question, John Perry, who's my epidemiological colleague, and ourselves asked the question, okay, well, you got to do a number of filtering steps to get to the stage, right? So what qualifies as a loss of function, this, that, what have you. And so if we make different filtering decisions, would we identify different genes, okay? And turns out we could. And so when we then did different filtering choices, we then identified two completely new genes, bassoon, okay, which I'm going to tell you about, and APBA1, which I won't talk to you guys about today. Now let me just explain what these stick and line and stick diagrams actually are. They're like a 6-dimensional figure, which for genetic nerds is all the rage. And so what happens here is that blue line, I can't get this straight. The blue line over here, there we go, is the transcript, okay? Just the transcript. All the introns have been spliced out. Now, where you meet a perpendicular line pointing up or pointing down, that's where there's a mutation that's been identified. If it's pointing above the line, it is increased body weight. Below the line is decreases body weight. The distance away from the transcript is the significance at the population level that is linked to obesity or body weight, and the size of the blob, the number of mutations we identified, 6 dimensions, okay, from there. Now, the one thing we want to get from here is that most of the perpendicular lines are pointing up. That's the point, all right? And so it increases body weight. But if you actually look at the effect size at the population level, now this is very, very rare. In 500,000 UK Biobank carriers, we found 65 carriers of protein truncating variants, which is what we used, okay? So stop gains, either through frame shifts or nonsense mutations, stop gains. And we find that it has a larger effect size than the MC4 receptor at the population level, so for body weight. It's the largest effect size we've seen at the population level today. Now, the other interesting thing about this is that it is associated with adult obesity. So UK Biobank is a cross-sectional study. All of the individuals, the 500,000 individuals, were recruited when they were between the ages of 45 and 60 years old. And that's fine. In fact, the only longitudinal thing that you study from UK Biobank is cancer. Okay? It's probably one of the largest ongoing cancer studies that are actually going on. But there is one question, okay, within you fill in your millions of questions, there's one question. And the question is regarding your childhood. It's at 10 years old, in fact, you can think about whether or not you know the answer to this. At 10 years old, were you below average weight, or plumper than average, the British study? Now, I don't know about you guys. I don't know much of what happened to me when I was 10 years old, but I did know that I was heavier than average because everyone told you, all right? And actually, when you did and went back and checked the validation for this, entirely correct. If MC4 receptor, just as an example, I told you 0.3%. If you actually look at the signal, big, large signal, I was larger as a kid because big kids, big adults. And most of the mutations within the leptin melanocortin pathway, big children, all right? Bassoon, not a sausage. 0 signal from adult obesity, from childhood obesity. It's an adult-specific onset obesity. Now, the interesting question, which we can't answer at the moment because of how rare it is, and the fact that it's only based on this one question, is when in adulthood. So I guess there are a few options. We could consider puberty. I think this is the least likely option because 10 years old is too close to you being able to get the puberty. But if it was puberty, that would imply hormones, I would have thought. You could then, well, did it occur at the freshers 14 or freshers 15 stage? Now that implies choice. So you've left home. Now you can go to the cafeteria, buy what you want, woo, and then you gain weight. That's a different mechanism because now it's feeding behavior question. Or is it middle-aged spread, okay? Because something just happened when I turned 50, whatever, okay? Now, each of those would have a slightly different mechanism. We don't know is the answer. But clearly, it's something has happened, some accrual of biology has happened, that you have really quite a large change in body weight, but specifically in adulthood, and no signal at all in childhood. And you can now see as well as the distribution. Now here is then the complexity and the slight nuance of it. So this is the distribution of body weight. The furthest, I can signal this. So the furthest right in terms of this graph, that is UK Biobank, the controls. So these are all the people, 400,000 or so, with no mutations in bassoon or MC4 receptor. And this is the distribution of body weight in the British population. Okay? The warmer colors are people who are overweight or have obesity. And so what this tells you is in the average British population, 2/3 of the population have obesity or are overweight. And I think the numbers are probably slightly higher here in the United States, but there we go. Okay. Now, this is where it's interesting. If you then take a look either at mutations in the MC4 receptor or bassoon, it increases your likelihood of being overweight or being obese, and that's fine, or having obesity, that's fine. But you can see that even with a mutation in the MC4 receptor, you can be of healthy body weight. And so when we first published that MC4 paper in 1998 now, we called it a number of different things. We called it a dominant mutation, okay? And we called it a monogenic cause of obesity, which are big words, all right? And dominant is after all, following the words of a 17th century Franciscan monk, okay? Big words. But if you look at this data here, now these are all loss of function MC4 mutations, but yet you can be normal body weight. So is it dominant? Right? I mean, what are we talking about here? So clearly there's background genetic information, there's a number of other things. And is it monogenic? So it clearly is a key genetic driver, but I just think we need to be more nuanced in our genetic terms, which we all learned when we were in high school. Dominant, Mendel's and his P's, but I think we do need to have a slightly more nuanced view of genetics. So, in conclusion, obesity is a chronic relapsing disease of the brain, and that's what it is. The pathways underpinning obesity are conserved through evolution. Using spatial transcriptomics in combination with NucSeq, we provide a detailed census of hypothalamic cells in a spatial context. And using this resource, we have identified new genes previously not linked to BMI. So this is so with that, I want to thank my senior colleagues, Steve Ratley, Tony Cole. The guys in bold have done the work. This is work that was done in close collaboration with my colleagues from the Max Planck Institute in Cologne, Jens Brunning, Lukas Stoinagel, my pharma colleagues from Novo Nordisk, but more importantly, the brain banks for which we can't actually get any of the work done, and this is the team. And if you can travel by then, we are organizing the 7th International Melanocortin meeting in Cambridge. And with that, ladies and gentlemen, I am very happy to take questions. Thank you so much.
Thank you. This was super interesting, and then I think I latched onto the point when you were talking about the cave fish, and you were talking about the fact that the cave fish had this, theoretical lack of preference for types of foods. They're just not picky.
Yes.
And I'm wondering whether or not you had any of the eating behavior or eating info about those people who have like bassoon, for instance, of whether or not these individuals are then also less picky about the foods that they eat. They have preferences, but they might have a wider swath of foods that they're willing to eat.
Okay, bassoon we don't know because they're rare, and UK Biobank doesn't have recall by genotype. However, certainly when it comes to mutations within the leptin-melanocortin pathway, then we do have data about this. Leptin deficiency in particular, so the first one which I didn't show you, is they don't have any preference for food at all. So in other words, because so I guess the analogy I will use is this. So because leptin deficiency means that you have no leptin, obviously. In a normal situation, because it reflects fat mass, you don't have leptin when you don't have fat. When don't you have fat? When you're starving. Okay. Now we know that we are. If you're really hungry, the simplest foods taste the best. Bit of bread, bit of rice, bit of cheese. If you're really, really full, you need to have desserts. Okay. You need to have something really, really. You become picky with your food. If you're starving, actually starving, like the kids with leptin deficiency and to a degree MC4 deficiency, you eat anything because you think you're starving. So you then lose the perception of it. And there are brain imaging studies, actually. So my colleagues, I'm not a brain imager, my colleagues have actually done this, where if you take people with no mutations in the pathway, and if you put them in fasted and therefore hungry and show pictures, then your brain will squash together pictures of dessert and pictures of broccoli, for example, all right, if you're really, really hungry. Whereas the moment you are full, then suddenly your brain only lights up with calorically dense food. If you take kids with leptin deficiency, they look like they're starving because their brain thinks they're starving even though they're very, very large. And even if they're fed, they have no discrimination between the two. But when given leptin, suddenly they're able to discriminate between energy-dense foods and energy-undense foods again. So within the leptin and melanocortin pathway, they don't discriminate between foods, no.
Oh, okay, cool. That's really interesting. Thank
Yep.
you.
All right, well, our next person comes up. I'll ask a question from online.
Yep.
Okay, right here. First question is, do you see epistatic feeding obesity growth effects among individuals with rare variants in different genes?
Okay, that's a good question. So the mutations in these genes are rare enough where if you have a mutation in the MC4 receptor, like a whacking graping mutation, you don't have mutations in other genes. We haven't found any in this particular pathway, any overlaps yet. Obviously, there are gonna be overlaps. But just in terms of, if I can add to that answer, what we then did was in our ALSPAC paper, the one where we looked at MC4 receptor, we then applied a polygenic risk score for regular BMI GWAS genes, so polygenic obesity. And what happens, we took the top decile and the bottom decile and then put against the prism of MC4 deficiency. And even with a monogenic condition, whatever that means, if you're in a top decile, you're heavier. If you're in a bottom decile, you are lighter, even with MC4 mutations. So your genetic background does matter, even when you have mutations within this particular pathway. I think it'll matter less with leptin deficiency, but that's so rare, it's difficult to be certain about. But yes, there are definitely, your background genetics matters.
So I was just thinking about, first of all, I really enjoyed your talk, it was really amazing. For the MC4 gene, has that been linked at all with like binge eating disorder, or how does binge eating disorder play into this larger picture?
So binge eating disorder is a very different thing. There was an early New England Journal thing about saying whether or not there's an increase in binge eating disorder. I think there probably is a slight overlap, but binge eating disorder is different from hyperphagia. So hyperphagia is a biological drive to eat because your brain thinks you are hungry or don't have enough fat. So therefore, it's a hunger-driven thing. Binging, I'm not an eating disorder person, but binging, I think you eat without being the hunger. So I think there are different drivers for the binging. There's a slight overlap with people in MC4 deficiency, but I think that is then a misdiagnosis of what binge, they think that the hyperphagia is binge eating. I think binge eating disorder is an eating disorder, which is the first part of bulimia, obviously, the binge eating and the purging. But I think that's a drive to eat that is non-homeostatically driven. It's driven for other reasons entirely. And just as an addendum to that question, because as a related question, when we first started studying the severe obesity, people then asked, was anorexia nervosa the opposite to severe obesity? And actually, it was a fair enough question. Turns out it's not. Healthy leanness, so people who are just congenitally just lean and annoyingly healthy, okay, is the opposite of obesity, okay, because if you look at it from a polygenic perspective, they have the lighter versions of the genes, whatever you want to call that. Anorexia, all of the signals which should tell the person suffering from the condition that they're starving, leptin levels are down, blah, 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 are all there. Where the anorexia comes from is the ability of the brain to ignore the signal. And so if you actually look at the genetics of anorexia, then what you see is an overlap, although not complete overlap, an overlap with obsessive compulsive disorder. So a requirement to control something within the context of anorexia, it's the food intake element, and so therefore you're able to actually ignore. So the eating disorders are now more of a psychiatric condition as opposed to a homeostatic condition. But this has only come out really in the last 10 years that we begin to know this.
I was, as a follow-up,
Please.
Do you find, or has it been found that there are a lot of people misdiagnosed with binge eating disorder when they actually have like hyperphagia or something?
I think probably, I don't know how many people, but based on this, because this New England paper that came up was quite a long time ago now, but I would say 15 years ago now, said that it was, that MC4 receptor is enriched for binge eating disorder. There is no evidence of that at all, given the fact that we found so many people. It's more the hyperphagia element to it. So I do think there is a misdiagnosis of binge eating versus hyperphagia, yes. Mm-hmm.
Okay, I'll ask another online question. First, fantastic talk. Are the MC3R or MC4R mutations associated with any other phenotype than puberty and body weight? The MC4R is widely expressed in the brain, and the MC3R only slightly less so. And a person who eats more might deliberate less about food choices than someone who eats less. Could there be an effect on executive functioning or impulsivity?
That's a lot of multi-part questions. The answer for the MC3 is I don't think it influences feeding behavior, actually, in terms of human beings. So let's put that aside. The MC4 receptor, I know that people who are actually studying it, I'm not going to answer the question because I don't know the answer specifically about whether or not it influences impulsivity. I think that actually if you consider the MC4 receptor is a genetic version of being hungrier, then I think you can probably sort of answer that question within the context of impulsivity. There are additional phenotypes to do with the MC4 receptor in particular, because the feeding behavior MC4 receptor phenotypes come from the hypothalamus, which is what I've actually showed you. But MC4 receptor is widely expressed throughout the brain. It also has an important effect on autonomic output. So what I mean by this, so for example, people with MC4R mutations actually have a lower given their obesity, lower blood pressure and lower resting heart rate. Okay, in fact, if you actually for their body weight, lower incidences of cardiovascular disease as well. Okay, in fact, if you wonder where would I find an enrichment for people with MC4R deficiency, given what we now know of everything, I think you need to find people who are tall, who are big, okay, very tall and very big. And actually, if you look within athletics, I think you can find people. NFL linemen, I bet you if I looked that we will probably go above 20% MC4R deficiency because you need to be large and not keel over because of a coronary. And these people have lower heart rates resting, and so therefore for that's the first thing. Sumo wrestlers probably I can think of as well. So big sports rather than fast. That's where I think we will find naturally occurring MC4R mutations.
Sounds like a cool study. I'm going to ask the last question.
Will the NFL fund me? I don't know.
Yeah, that's the important question.
This is right, yeah.
Okay. There's a question as to whether any of the obesity genes known play a role in surviving starvation or severe malnutrition.
Good question. I don't know the answer to that. I do know, for example, for specific diseases, slightly tangential, that if you look at there are certain diseases, Huntington's disease, for example, rare situation where you end up with neurodegeneration, where you do a lot better if you start at a higher body weight. Now, I think it's more simple. It's more complex than simply having a larger reservoir. So you have a larger reservoir of energy, so therefore you do better and survive longer. I think it's more complex than that. And people are trying to understand, well, hang on a second, why do these people who are larger do better when they actually hit something like neurodegeneration? So that's not exactly the question that's being asked, but I think there is likely to be maybe not pathway direct elements that are there, but we can take from the Huntington study whether or not this might be the case.
And so we're out of time, so I just want to thank one more time our speaker today.
Thanks, folks.